And good evening, everybody, and welcome to Pint of Science Netherlands Taus episode 5. My name is Hannah. I am a research assistant at the Prinses Maxima in Utrecht, and I'm a Pint of Science volunteer. On this side, we have Jeanette.
Hi, good evening. My name is Jeanette. I'm a science communication manager at the Radboud University Medical Center in Nijmegen, and also the communications officer of this Pint of Science Netherlands team. And we have a great guest tonight who's going to tell us about snakes.
Yes, so we're going to be looking at snake venom organoids this evening. And no, it's not the next Alien film, as many people have asked me. But to introduce the topic, we're going to show a short video that has been pre-prepared for us, just to let you know a little bit what are snake organoids.
Wait.
Can you hear it? I can't hear it. Margarita, can we start again? Sorry guys, we're having a few technical issues. It doesn't seem we can get the sound. We'll try and start the video again to see if we can get the sound going. So unfortunately, we weren't able to get the sound on that one, but it looked very interesting. So instead, we're going to have, which we were going to next anyway. So tonight we have Jens Puschhof from the Hubrecht Institute will come in and tell us everything that we couldn't hear during the YouTube thing. Yeah. So Jens, please welcome to the studio.
Hi. So pleasure to be here. I think it's the video was full of spoilers anyway, so thanks for talking.
And now there's even more mystery. What was that actually about? So well, I guess
theory. Absolutely. So you've seen a bit of that already in the video, and now you get the narration from me. So I brought a couple of slides for that. I probably can quickly let me know when you have the slides on the screen.
Yes, they're here.
All right, perfect. So yeah, you've seen those faces in the video already. We're a team of 3 PhD students doing this research together. So that's Jorik Post and Hugh Boehmer and myself. And this was all about growing mini organs off the snake venom gland. So we got our snakes ready for a pint of science today. But to give you a start, I want to go into that word, organoids. And that's something which has actually been developed in the Netherlands about 10 years ago. And this gives us the ability to grow tissues from outside of our body in the lab. And all of this is based on stem cells. That's our boss, that's Hans Klevers, who's working here with us in the Hubrecht Institute in Utrecht. And he made a big discovery, I think like 13 years ago. And that's that we don't only have stem cells in embryos, but even in adults. So in yourself, you have a lot of specialized stem cells in most of your organs. And those have the task to regenerate an organ. And when you have like an injury, they will lead to repair of that organ. What's really cool and what's the basis of organoids, or these mini organs, is that if we take these so-called adult stem cells out of an organ, they do exactly what they should do in a petri dish. So in the right conditions, such a stem cell will feel that it's like outside of its environment, and it will start regenerating the organ it's supposed to make. So it will divide, it will make more cells, and those self-organize into a whole mini organ, so-called organoid. In this case here, that is the intestine. And you see this is about a 2-week time course, and at the end, we have a mini intestine. That, of course, is an animation video, and this is microscopy. So that's what we see every day in the lab. Over a couple of days, a mini organ growing. In that case here, that is an intestine.
So Jens, you have really like small intestines growing in a little dish in your laboratory.
Absolutely. So I think about 60 people, and everyone has their own mini organ type growing every day.
And these are intestines?
Yes, these are actually intestines from the mouse.
Okay.
Wow.
Yeah. And there's a lot of applications. So we're really here talking about our snake research, but there's many things we can do now, like testing if drugs work, regenerating tissue, and really understanding how different organs in the human body work by growing them in the laboratory. But what we set out to do is see if this would also work for the snake. And here I'm listing the motivations we had. The first one is that in the last decade we've learned a lot about all of our different organs through organoid technology, but what hadn't been done was growing them from a reptile. And so as biologists, we're really interested in seeing how does the thing work in human, but how does it work in all the different animals as well to understand it better. The second, also as biologists, we need model systems, like when we want to develop a new cancer drug. We can do this nowadays with these mini organs from a tumor, like having a mini tumor and seeing which drugs kills the mini tumor. Because it would be very tricky, it would be unethical to try all of that in patients, so we need an easier system to test that. And the same situation applies to snake research. We can't fly to Australia every time we have a great idea, like catch 10 snakes and then try it out. It would be much better to have something in the lab where we can test our ideas and understand how snakes make venom. And that's what we also wanted to do, making snake venom gland organoids. Probably the most important reason is that snakebite is a huge global health problem. There's more than 100,000 people dying every year because of snakebite. And for the most common treatment with antivenom, you still need the original snake venom to make it. So what we were hoping to achieve is that when we can make this venom in our mini organs, it would be a much easier way and a much more controlled way to harvest it, to help this process of making antivenom to help snakebite victims.
So
and finally,
are you growing snakes in a dish or just the venom glands?
Exactly. So we tried a lot of different organs, like just to see, because we can grow basically any human organ. So we also wanted to see if that works for snake. We could grow a variety, but our favorite by far is the venom gland, just because venom is the thing we're really interested in. And the fourth reason why we're really interested in venom is that it's a mixture of compounds, and it has really crazy effects on the body, like a kind of paralyzing effect, or it can make your blood pressure drop so you faint at the spot. And normally these things are really bad, like that's how a snake kills its prey. But on the other hand, they can be very beneficial in some conditions. If you have high blood pressure, probably you want something to decrease that just a bit. And so that was also thought of us to see, can we probably make some of these toxins in our culture and help making new drugs for some human diseases. So we had a lot of good reasons in mind to kick off this project, but in the Netherlands we encountered a big problem. That's how do we get a snake to our lab in the Netherlands? And luckily, Joep and Jorik, they're both Dutch, so they knew this TV guy you have here. Freek Vonk was going to the jungle, catching snakes. So we thought, why don't we write Freek an email and say, hey, can you help us get a snake for this project? And it turns out he's actually a really good biologist. So he did his PhD some 10 years ago in Leiden with Mike Richardson, who also became a great collaborator. Yeah, and over the past years, they have been really great in assembling a network of breeders and of zoos. So whenever a snake is dying in a zoo, they get a phone call, or we get a phone call to go over there and get some of the tissue of the venom gland to try to make organoids. Yeah, and that's our zoo so far. We've got organoids from 9 different snake species. And you see the snakes are really colorful. The organoids are a little bit less so. So on our standard microscopes, they really look a little bit like balloons of like a cell layer with a hollow inside. But that's when we teamed up also with Hannah's lab. That's Anne Rios at the Prinses Maxima Center here in Utrecht, and Ravian, who did a lot of this imaging. And so we can get colors to the structures of these cells. And now what we can do for the first time is flying through such a mini venom gland. And you do see everything in orange here is a nucleus of a cell, so the core of a cell. And we have a lot of other proteins labeled here. And you do see that there's a hollow inside of this mini snake venom gland. When we think about what a venom gland normally
does, like a little ball, then we've just gone inside.
Exactly, it's like a balloon. So there's a layer on the outside, and this layer is made up of the cells, and the inside is hollow. Well, at least it looks very hollow here. But in the normal venom gland, you do have the toxins in there. So the snake produces toxins all day long, and when it bites the victim, then it wants to release all of these toxins at once. So we're wondering, do the organoids make the same? Do they like collect all the venom in the inside to probably release it with some tricks? So for that, we zoomed in one level further. So this is transmission electron microscopy, where we now can look inside individual cells. So this one here on the outside you see is again a core of a cell, a nucleus. But it's really cool towards the inside. You have these round structures which are filled with something dark. And this here is the inside of an organoid, and it also has this dark filling. So we really started to wonder, is that venom which the snake venom organoids fill their inside with? And to test if the filling is actually doing the same as venom would do, we teamed up with the team again in Leiden with Mimetas, a biotech company. And this is really cool to see, like in the past 10 years, what has been developed. So when a snake bites a victim, it can, for example, have a paralyzing effect on the muscles. So it stops your muscles from working, you cannot run away, the snake can eat you. Probably 10 years ago, we would have had to take a snake and take a mouse and have the snake bite the mouse to see what's happening. But now we can do all of this in a petri dish. So we developed the snake venom gland in a petri dish, and the company Mimetas, they have muscles in a dish. So these are muscle fibers grown on a chip. And what we can test now is, does the venom also block these muscle cells as it would do in nature? So normally muscle cells communicate in these wave movements. In this case, this is calcium that they transmit to each other. And you see these waves moving there. And very interestingly, when we add the toxins from our snake venom gland organoids, this is pretty much completely stopped. Just to show, when we collect the venom from our organoids, it can do the same thing which it would do in nature. It blocks the firing of muscle cells. It is paralyzing these muscle cells on a chip.
So just to recap a little bit, you get a bit of tissue from a dying snake in the zoo. You grow it in a petri dish. It produces toxins, and then you put that on muscles in a little dish, and then you see the effect.
Exactly, that's how it works.
Wow, it's amazing. Yeah. I think this is amazing.
Yeah, that was really cool to see that we can really make the real thing, like the real venom, to have these effects. Yeah, and that's already my last slide. Just the key takeaways. So starting with organoids, which are these mini organs, which the lab of Hans Klevers has developed in the last decades. And what we could do now, we actually just published this research this year, is taking this technology further towards reptiles, starting with a lot of different snake species, and these organoids actually make venom. And when you think about our motivation, find new drugs, make antivenom, understand how snakes make venom, this is a great starting point. This is not the finish of a research project, but this is really the start of trying to see, can we make antivenoms Are they in the end cheaper? Can we make more? All of this is still up to be answered, but we're really happy that we have now such a system as scientists to start exploring all of that. Yeah, and just again to give credit to the team, you see Joep and Jorik over here, Hans Klevers, our boss, but all these names here, they're coming from all over the world. They're specialists. Some of them are snake researchers, others are microscopy specialists. I think it really highlights how much of teamwork science is to make such a project work, and really everybody contributing something to get all of these different measurements to really understand what kind of organoids we can make there.
It's very cool.
Right.
Thank you for that explanation.
That's it for my slides.
So I guess my first question is, as you said, we're in the Netherlands and you don't have a lot of access to venomous snakes here, as far as I'm concerned, when I moved here. So what on earth prompted you guys to start a snake venom project, and how on earth did you ask your boss, if that's okay?
There's many different stories circulating about that, because that's something, yeah, which we do get asked a lot. I do think it comes from this the technology that we can grow these organs, we can make mini organs of so many human organs. So it was a little bit, you know, like us thinking, what can we do? What's still out there, which would be really cool if it works? And yeah, so Jorik and someone said, yeah, well, snake venom and organs, that's such a cool organ, because people think a lot about snakes. It's just fascinating animals, and they have a really clear task, these cells, making venom. So also for us to see, is a project successful? Are we really making the real thing? That's something we can easily understand. If you want to grow a brain, people do that, and it's amazing, but brains are so complicated. Whereas for us, it's really, if they make venom, they're a cool thing. There was a little bit a mixture of things, and of course coupled to these 4 motivations of really helping a global health problem with snakebite and making biology, like understanding biology of snakes much better. So I think mixture of all of that.
Great. So maybe we should say that if people have questions for you, they can type them in either on Facebook or on the YouTube chat, and then we will forward them to Jens. And while people can think of their question, I'll just point out that Jens is actually sitting in his laboratory in Utrecht, and in the background you can see some rubber snakes. So before the serious questions, what are these snakes doing there behind you?
Yeah, I'm a little bit embarrassed you pointed it out. This was a bit of an Easter egg for Joep and Jorik. We had these rubber snakes accompanying us throughout the project. Yeah, and I thought that was a nice decoration.
Yeah, it's your mascot in the lab. That's very nice. So have any anti-venom, has that been produced yet in the lab, or how far are you still away from the application or having a real treatment from this?
Yeah, so we haven't made antivenom yet with these organoids. It really shows that you need a lot of expertise to get such a breakthrough from the start to end, to really reaching the patient. That's happening right now. Right now, it's a lot about scaling, like making much more venom, because we're basic scientists. We get really excited when we have like a couple of grams, a couple of million of these cells, but to really make large amounts of venom, you just need to scale that. So we're working with people in America and Boston. They're trying to scale up this technology. And we have collaborators in Liverpool, Nick Casewell, who then are trying to make antivenom. So that's all ongoing. We don't have the answer yet, but we're really trying hard, because, of course, to make a real world impact with that, you got to go the extra mile. Yeah, so then I can imagine like whole factories where there's like little dishes of like snake organs growing, and then exactly. That's what I'm doing as well. You know, there was a biotech company in California housing 2,000 snakes in its basement just to like make venom for clinical trials. So I think if they can do it with snakes, probably somebody can do it with organoids. So that's what we're trying to achieve in the midterm. That's all not happening overnight, but yeah, we're trying. So you're actually saving also a lot of snakes from living in factories, right? Yeah, we hope so. Of course, the technology needs to be completely developed, but I think that's a great thing about organoids. Like as with the example with these mouse muscles in a dish, yeah, in the end, you start with animal tissue, but if it works well, you can really reduce the use of animals. There's some experiments where you will always need it, where it's about the whole body response, but there's a lot of things which we can already do with organoids. I think it's really cool. You need an animal in the beginning, but at some point the system is so good that you can really do very meaningful science with it. So yeah, I like that angle a lot. And one thing you hear about antivenom is obviously the cost of it, because housing all these snakes and the danger behind it. Would one of the aims be, do you think, that this could help reduce some of the cost of antivenoms in the future? It's a very good point, and that's one where we don't have a good answer to. To be completely honest, organ technology is not super cheap. So that's why we're also working with our collaborators in America to get these organoids on other platforms where we save materials costs, really make it economic. Like, as a scientist, you don't really think a lot about like the economic perspective, how much these things cost. But yeah, we start thinking about that and see if we find solutions. In the end, I think a big argument for this will be the quality of what we get, that we really have a very controlled system, making venom in a very controlled way. And in the end, if you want to go to a patient, you need this control. You need to be sure what you're giving a patient is exactly what you want it to be. So I do think with all of this, we have a good angle to develop this technology. If in the end the price will be lower, it is hard to predict. We really have a lot of work to do to make this a cheap alternative.
Yeah, great. So we have had one question from the audience. I can bring up the question now. So this is from Ravi van Inneveld. So if snake organoids make venom, what do intestinal organoids make? As you mentioned, you have some of those in your lab.
Yeah, oh, that's a tough one because I said the snake venom is a great organ because we really know what it's supposed to make. The intestine has a lot of different cell types. So there's some which make the mucus, like the slimy substance lining your intestinal surface. There's others which, like my colleague Joop on the snake team and I are really interested in. These are the hormone-producing cells, which like secrete hormones onto your body to tell your body like what's inside of the gut. Yeah, and intestine has a lot of other functions. It takes up food. So yeah, I could talk another half an hour about what the intestine makes. I think I'll stop here.
So it's not if you feed them, then we get some waste out the other side. That hasn't happened yet.
No, but that's really cool. We should have more digestive organoids. It's a good one.
So we've had
another question.
Oh, yeah.
Sorry. Let's see if we can also show it here. It's from Sulov. How many days does it take to develop venom in vitro and in vivo? Maybe we should explain this. So in vivo, that means in live organs, right? In the real system. And in vitro means like in the Petri dish, as you described in the laboratory. So is there a difference between those two situations?
Yeah, it's a really good question. We always have to compare. Like, we want to model the real thing with our organoids. There's data out there for the actual snake, and I think about 4 days after it releases its venom, then you have like the peak of when it's making a lot of new venom. So again, it is filling like the middle of the venom at some point is filled with venom. So I think they also stop at some point, like because they're full, and when they bite, they release the venom, and then they have to start making it again. For the organoids, which is another really cool thing about organoids, we have all the control. So we have a so-called stem cell medium where we expand the organoids. We can start from one stem cell, and in the end, we have billions of cells. But when we want to make venom, we do something called differentiation. So that we don't want the stem cells, because stem cells are really busy with like regenerating an organ. The cells which make the venom are a little bit more, they sit around, they don't divide much. They just make venom. So we can trigger that process. And from then on, it takes, I think, like about 10 days to have really good venom amounts. They start very soon. So I think after 2 days after we start this process, they already make good amounts of venom. But we normally wait about 10 days to really harvest the venom. So in the end, I think it's roughly comparable. Like the peak is probably after 4 or 5 days. They fill, and yeah, then we harvest the venom.
Oh, very interesting. So you've had one more question again from Facebook, which we can bring up. So Andrea, she really likes your talk and very nice imaging. Yep. And she said, are you growing other organoids from snakes? So you mentioned that you tried a few different things, but what else were you able to grow, and where did that lead?
Yeah, so of course, intestine is a very, very good guess because snakes have really fascinating biology there, especially constrictor snakes, but also venomous snakes, not eating for a long time. And then they catch a huge like pig and they devour it, like, probably in these pictures of a snake where you really see like the bulge of a large animal there. And then they have to ramp up all the physiology. So we're trying to study that. We're trying to get the intestines to grow. This is really ongoing work. Yeah, I think we got some kidneys to grow. We tried something called the vomeronasal organ, which is a really complicated word, which is a little bit like somewhat compared to our nose, like or to our taste organs, to the tongue, where the snake is sensing like chemicals from its environment. So we tried to get that to grow and doing a bit with that. But I really have to say the venom guns are like our favorite organoid, where we can do a lot of things with. But we're trying to expand this and find some other cool parts of biology.
So would it? Would it technically be possible to grow an entire snake in your Petri dish?
I think
That's too science fiction.
at some point it would just start being a snake again. Like in the end, it's just like then you have a whole animal again that's like a little bit away from the organoid. One thing I'm personally very interested in are so-called co-cultures, where we bring different types of cells together and see how they interact. So I do think it's a concept of science that we like take things apart and we reassemble them and we see what happens. So I do think like getting all the different adult stem cells, growing all the different organoids and bringing them together to see like how does venom gland interact with the muscle right next to it? Like when the muscle is squeezing it, pushing the venom out, how does that affect how new venom is being made? And all of these things, I think we can build. It's not always easy, but that's definitely something we can do. If at some point we have an entire snake again, I think we can approximate it. Yeah, I guess that's my best answer.
So we don't need to be afraid of snakes coming out of the Hubrecht Institute yet.
No, I think there was like a featured on IFL Science on this Facebook page, and there were really people speculating wildly like about the bad impact of people making toxins.
Come back. Forgot the electric cultures in the incubator. Oh no.
So far, nothing terrible has happened from this project, so I think we're good so far.
Yeah, okay. Good to know. Just going back to the organoids, so you said you had 9 different species of snakes at the moment. So do you have a goal of what you want to get in your kind of, in your storage of how many different, because as we know, there are hundreds and hundreds of different types of venomous snakes out there. So, where are you guys aiming towards?
Yeah, that's actually one of the big things happening right now. So Freek Vonk, you saw on one slide, he's just become a professor or group leader in Amsterdam. And we're working a lot with him now to expand this technology, to go to about 50 species, also to really conserve a little bit of the biodiversity on the organoid level, because it's not so well understood, all the different toxins like which the snakes can make. And these organoids, we can keep them basically forever. Like we haven't found a point where they would stop. We can freeze them, we can thaw them, and they're alive again. They can make all these toxins. So yeah, Freek just has a first PhD student, Matthias Bittenbinder, and he's a lot in the Hubrecht these days. Yeah, bringing snakes, trying to get further. So I think we're a little bit further than 9 already, but I didn't have nice pictures for those. It's happening. I think we're really moving towards that. And there's so many cool species which we haven't covered yet. So that's much more than just this one snake venom gland organoid, but all the different types of snakes, the different toxins they make. Yeah, lots to be discovered. More work than we can do right now.
Do you find a big difference between the different species, and have you found any correlation to where the different species are from, that the toxins are similar?
Mm-hmm. That's a really good question. There's a lot of variation known from nature that like different species make very different toxins. There's very different types of toxins. Some block your nerves from firing, some lower your blood pressure. So, and this is just 2 examples. Some attack your heart. So it's really a lot of a variety of mechanisms. And it even varies between snakes of the same species. So 2 different individuals can make quite different toxins. And even one individual snake over its lifetime can change it. So when you're a baby snake, probably you want to prey on insects or on lizards. And so you want toxins which kill those. And when you grow big, you probably want to eat mice or something. So you change your toxin mixture. And yeah, we're starting to study that with organoids as well, like how these switches happens, how a cell decides which of these toxins to make. And it's a lot of toxins, a lot of snakes, and we're just...
Oh, that's very interesting.
Yes, I can imagine that there's a lot of chemistry involved in this research as well, right? It's not just the growing of the cells, but also analyzing all the substances. I guess you're a biologist by training? Or?
Exactly. So I'm somewhere coming from the cancer research direction. I'm still doing cancer research as well. So this is really cool research, but we're also doing other science. I'm definitely not a chemist, and that's really on point. Part of that team around Jeroen Kool in Amsterdam, they're really great venom chemists. So when we first had this like filling of the organoids, we thought, okay, this looks great. That's probably venom, but how do you prove it? So then we got in contact with them and we started collaborating, and they can really measure this. Like they find out all these small proteins, what they are exactly, and what toxins they make. So that's really valuable, and yeah, so much to learn with them from these collaborations.
Yeah, and a lot of science coming at one point, right? Different fields emerging. Yeah, great.
It's been really interesting. just coming up to time, just about. So is there anything else you kind of wanted to add about your organoids and where you guys are going?
I think you covered that really well. I guess these were really, really good questions you all had. Yeah, probably just want to say hi to Jupp and Jorik if they're watching. I'm not sure if they're watching, but great fun working with you guys. Oh, yeah. And also the rest of the team, like so many people. Yeah, it was great fun working with all of you.
Oh,
yeah, and the video that we wanted to show that you couldn't hear the sound of, in the comments you can see the link, so you can just watch it yourself, and then you can have a look. Maybe a final question from Ravian that just came in before we close, because I think it's an important one. As the toxin is harmful to the cells, how do the organoids protect themselves? I think that's a very important question. Why does the snake not kill himself with this organ, with the venom?
Yeah, also really good question. Many different answers to that. The most convenient answer for us was we were lucky, or not lucky, but we chose a neurotoxic snake, which really makes a lot of like nerve toxins. And these organoids, they don't contain nerves, so they're really just the venom-producing cells and the stem cells. So if something is blocking the firing of your muscles, you're pretty well off if you don't have a muscle. So that, I think, made it easy to grow those. There's others which are called cytotoxins, so these are cell-killing toxins. And for them, we find that these cells may make some components which protect themselves. So they not only make the toxins, but they may also make things which block the toxin as long as it's in the gland. And these are just two mechanisms. There are probably a couple more we could go into, but as a general answer, either some of these organoids don't make the toxins which would kill themselves, or they make some protective factors themselves. And once they secrete that, probably like the pH, so the acidity changes, and suddenly the toxin becomes active. But this goes into the realm of speculation towards the end. So yeah, but these are just two mechanisms.
I think we could keep talking about this all night.
Yeah, exactly.
I certainly could. I'm really happy if you're around.
Yeah.
Thank you so much, Jens. I found it very interesting. I think it's fascinating work that you're doing, and indeed very beautiful images you're making.
Yeah.
Well, thanks for that. Thanks, Anna. Thanks for having me.
No problem. And to all our viewers, you can follow us at Pint of Science Netherlands, and there we'll put up some links. Again, we'll put up the link to the YouTube video that you weren't able to hear, and on our link tree that we'll also share, you can see some links to some of the publications about Jens and his lab and the topic they've been working on, and so you can kind of follow the story and see where it goes over time, which I think is really interesting.
Yes, and we're taking a little bit of a summer break now, but we're back with these Pint Thuis webinars or meetings. So the next one is on the 23rd of September, again, Wednesday evening at 8 p.m. And a very different topic. It's on the science of art conservation. Sorry. We have chemist Katrien Keune, and she is the head of science at the Rijksmuseum, the big museum in Amsterdam. And she will tell us, about all the science of conserving art. So that are two different fields coming together. Very interesting. So the 23rd of September, we'll be back here.
Okay, great. Right. And thank you, everybody. Enjoy your summer and have a lovely break. And we'll see you back in September to learn about art.
And cheers.
Bye. Cheers.
Cheers.
Oh, quick.
Cheers, guys.
Bye, everyone.